Hello, welcome to single-cell multiome analysis series with SciDAP. In the previous video, we were reviewing the output of the step 6 ATAC clustering analysis. And now we will move to the next step, which called WNN clustering. The weighted nearest neighbor method integrates multiple single-cell modalities like RNA and ATAC by learning how much each modality contributes to distinguishing a cell from its neighbors. We will call our new sample Step 7, WNN cluster analysis, and we select the last samples that we created. It's our Step 6. Then we select our aggregated sample. Dimensionality directly changes how much structure each modality contributes to the joint graph. More dimensions let that modality encode more variation, increasing its influence, while fewer dimensions reduce its weights on the final integration. I will try 50-50, making it equal. And we will do 3 clustering resolutions as we did previously. We could also select gene markers and peak markers that will help us to annotate the clusters and identify cell types. The genes of interest to highlight. You probably remember from RNA step this GZA optional analysis for gene sets. I think I will skip this for now. This time I will select Save Row Counts to Loop file. Then we could try to use the loop browser later. Then just click Save. Now I would like to run one more sample in parallel. Remember we had some issues with the silhouette scores in ATAC? I think it would be reasonable to try giving the ATAC part less contribution and rely mostly on RNA part for the generation of the final WNN UMAP. In the next video, we will see the final clustering result. Thanks for watching.